Hello, and welcome to our uh, prokaryotic features portion of our lecture in our cell modules, cell structure and function modules. Um, the best thing to take away from this is that there is no nucleus in a bacterial cell. There is only a nucleoid region. Okay, that nucleoid region is about where all of the DNA is, and inside you have these naps that kind of help condense the DNA. In addition to bacterial DNA in their nucleoid region, remember that, you can have plasmids. Plasmids are really important in molecular biology, like for someone like me, where you want to put in genes of interest into things, but also these plasmids lend themselves to have antibiotic resistance. So these are often passed through the sex pilus of prokaryotes, which let them get these nice antibiotic resistances, and that is why you don't want to skimp on your antibiotics. You want to complete them all the way through so you don't leave any stragglers who can confer resistance to others. Um, Bacterial cells and eukaryotic cells, so both prokaryotes and eukaryotes, have ribosomes. They are living creatures, so they have ribosomes. Ribosomes are there to create protein. You need protein to do things. You need protein to make the cytoskeleton here. You need protein to make almost everything you see in this picture. Um, and those proteins are made with ribosomes. In the prokaryote, you have a 70S ribosome, and in the eukaryotes, you have a 80S ribosome. Why is that useful knowledge? Well, it means that we can attack the genes that create a 70S protein versus an 80S protein, and we can probably leave our host who's infected unaffected by our drug, by our treatment. So we see a difference between them, and we know that prokaryotes and eukaryotes are different right here in the ribosomes, even though they both have them. Okay, and of note, the ribosomes in prokaryotes look a lot like the ribosomes in mitochondria, all the way up there in the corner. We've talked about this in the lab, how we're going to do it in the lab. We've talked about spores, with how we need the autoclave to destroy them, how they can, because they can survive extremes. They could survive temperature, even radiation, so we need to have a combination of like pressure and temperature, or radiation and sterilization with chemicals. We need to get rid of these spores, you need to take proactive action. You can't just let them stay there. So remember, they are vegetative structures. Uh, they are survival structures. They are not vegetative cells. The vegetative cells will sense a lack of nutrition or resources, and after some time they will begin to sporulate and make a spore. And we will actually see these in the lab. Again, all prokaryotes and eukaryotes have a cell membrane. Okay, the cell membrane is universal. Everything needs it except for viruses. And it is known as a plasma membrane. And note all of our friendly phospholipids there. Phospholipids. Okay, and look at the outside of the cell here. The extracellular matrix is not composed of phospholipids, right? It's composed of glycoproteins and other things, not phospholipids, okay? The cell membrane offers a selective permeability where they can allow certain things through and not others. And because of the distribution of protein and cholesterol and phospholipids, it kind of tends to stay fluid and never kind of in one spot. And this lends itself to what's known as the fluid mosaic model. Interestingly, the amount of cholesterol in those yellow things, the amount of cholesterol on your cell membrane lends itself to being more fluid or rigid. So more cholesterol, more fluid; less cholesterol, less fluid. As we're talking about the plasma membrane, it's going to be important to talk about membrane transport mechanisms. So it's kind of a segue in um, uh, our partitions of prokaryotes, but we kind of need to mention these membrane transport mechanisms in order to uh, understand what it is we mean by selective permeability. Okay, so if you are a small enough solute, you just kind of go through passive transport if there's two little salts on the other side, and you just kind of wiggle your way through the phospholipid membrane. You will go from an area of higher concentration to lower concentration. Okay. You can have charged particles, and you can have facilitated diffusion. Things that would naturally go through um, are able to go through channels. Okay, and those channels typically would only be big enough to allow what it wants through. But it doesn't require any energy because you are still using a gradient. It's just too big to get through the um, phospholipid bilayer. And then finally you have active transport right here. And active transport obviously requires energy, active energy. And that's that big yellow blast right there on the screen. And this generates ATP. And so ATP is what we get energy. ATP is what we'll learn in our metabolism lectures. So keep in mind what ATP is, and that it basically powers all of our cell reactions. There's some other things that do that too, but ATP is the main one. All right, back on track. We get to our cell wall. The cell wall we've talked about at length with the gram stain. Okay, so remember peptidoglycan with our gram stain. Peptidoglycan is the main component of a cell wall, and if we can attack how this is made, or we can attack it itself, we can destroy bacteria again without destroying the host. Okay, and that is dependent upon it getting through the gram-negative outer membrane. Okay, remember the cell wall keeps its shape when it's desiccating. In our osmosis section, it would cause plasmolysis if it shrinks upon itself inside the cell wall, and it could cause a nice turgor pressure before bursting if inside the cell wall as well. Okay, so here we are, our link to the future, where we'll see that we can attack various parts of bacteria that do not necessarily attack humans, but like DNA synthesis does. So we are hesitant to prescribe those except when we actually need to. All right, and then obviously no archaea causes disease, but no archaea contain peptidoglycan. Cell wall would like to stress this before we pass on to the next video. And we see here in gram negative cells this LPS layer, okay, lipopolysaccharide layer. This lipopolysaccharide forms an endotoxin, okay? It forms an endotoxin to humans, and so if you kill this bacteria too fast, if you have an infection, you can get septic shock, you can get fever, and it can cause a bunch of problems. So when you are infected with something gram negative, it's not so easy as let's just destroy it. So kind of keep that in mind. And the key word here is LPS, lipopolysaccharide, and endotoxin. Endotoxin should probably be bolded. All right, and with that, we will pick back up with glycocalcises.